Hi, everyone. Thanks for the opportunity to speak. So as an introduction of sorts, maybe for those who are less familiar with the field of tuberculosis diagnostics, some of you know it as the more accomplished, more handsome Hemsworth brother. It's a field that, like many other scientific fields, has seen a gradual transition from more traditional phenotype-based approaches towards genotypic diagnostic methodologies that aim for higher accuracy and higher throughput. Both of those, as I'm sure you'll agree, critical from a public health perspective. Among these NGS tools, there are two that have gained significant traction in research settings and now are starting to be implemented at a larger scale. These would be whole genome sequencing, which focuses on a comprehensive analysis of the entire genome. And on the other hand, we have targeted next-generation sequencing, which instead centers its sequencing efforts on a few genomic regions that can be considered of interest. The purpose of this talk is going to provide a brief overview on the differences and similarities between these two, both in terms of approach and performance. So for the purpose of our comparison, the MAGMA pipeline, which was developed at the University of Antwerp, was used as an example of the WGS approach, while the dplex MIG-TV assay was used to evaluate the capabilities of the TNGS technology. And some differences between the two can be spotted right from the start. So for example, in terms of input sample, dplex requires a minimum batch size of 20 samples, but with a huge advantage of being able to sequence directly on sputum. MAGMA, on the other hand, still requires a culture step, but in turn is able to handle batch sizes of as low as 4 samples. In terms of the actual analysis, dplex, as we've said, focuses on a few genomic regions, which amount to a total of 13,000 base pairs. By thoroughly analyzing these regions, dplex is able to detect variants at frequencies that go as low as 1 to 3% and provide resistance information on a fixed panel of 15 drugs. MAGMA, on the other hand, analyzes the entirety of the MTB genome, which is around 4 million base pairs. Thanks to this, it's able to produce resistance information on a panel that right now comprises 23 drugs, but that can be easily adapted for new genomic targets and eventually new drugs. And as for the limit of variant detection, it's comparable to dplex as long as similar sequencing depths are achieved. And finally, two differences in regards to the phylogenetic type and capabilities of both tools. Of course, dplex wasn't designed for this in the first place, so the outputs that it's able to generate in terms of phylogenetic trees and its ability to infer possible transmission events, it's unfortunately a bit more limited. I would like to briefly elaborate on two of these differences. The first one is the importance of dplex's ability to avoid a culture step. By doing this, it's able to avoid the issues commonly associated with culture, which are very much present in WGS technologies such as MAGMA, and that include the possibility of diversity loss in the sample, but also significantly increased turnaround times, especially in a slow-growing bacteria like TB. And they also entail significantly more stringent requirements in terms of infrastructure and biosafety. The other important difference is the comprehensiveness of both approaches, and this can be easily illustrated by how they infer drug resistance. So what we see here is the 18 gene panel that dplex analyzes in order to produce a resistance output for 14 different drugs. Meanwhile, these are actually the candidate genes listed by the WHO as potentially being involved in drug resistance acquisition. And then we have MAGMA, which of course is able to analyze all of these genes, as well as additional ones that enable it to generate resistance information on 7 more drugs. What this means is that the resistance calling output for MAGMA is significantly more extensive and more thorough, and more importantly, it allows MAGMA to offer a wider range of possible treatment when resistances to common antibiotics are found. Similarly, as we've mentioned before, the magma's ability to analyze all potential lineage markers in a sample allow it to provide an accurate phylogenetic output for a cohort of samples, while dplex is restricted to a few SNPs and a polytype for any given sample. So these are the differences on paper, but to actually test the performance of these two approaches in the field, we designed a comparison study. For this, we collected sputum samples from 67 consecutive patients in a clinical trial here in South Africa, which had been diagnosed with rifampicin-resistant TB by Expert Ultra. The dplex assay was performed on DNA extracted from decontaminated sputum sediment, while WGS data was obtained from DNA extracted from liquid cultures of the same sputum sediment. And the first place where we looked for differences were the yields. So is one of the methods significantly more robust when it comes to dealing with clinical samples? The differences we found are not statistically significant, but we think that they bear some clinical relevance. Magma is essentially able to provide a resistance output for 10% more samples, which means that 10% more patients get a resistance profile and consequently a treatment. And from the ones successfully analyzed by dplex, a few of them had what dplex labels as a poor sequencing score, which means that actually not all of the targets that dplex wants to analyze are successfully covered. For the samples where an output from both methodologies was available, we then focused on differences in resistance calling. And here we found that the outputs for both dplex and magma are largely similar. Only in 10 samples we found differences in the resistance profile, and only one of those samples had a significantly different resistance profile. And if we focus on individual variants, the differences are even less, with the vast majority of individual resistance goal being identical. We did find some differences, of course, and these were due to a number of factors, such as the detection of minor variants, so variants that are present in a frequency below 10%, instances where a variant is detected by both methods, but its impact on drug resistance is classified differently, and then a few instances where a mutation that is deemed by magma to be relevant towards drug resistance simply falls outside the targets that are covered by dplex. The case of minor variants is particularly interesting because we found a surprising level of disagreement between dplex and magma here, despite the overall agreement in overall resistance calling. So what you can see here are not differences in frequencies, but rather instances where a mutation that has relevance towards drug resistance is found by one of the approaches and completely missed by the other. I'm going to be honest, I'm not really sure what that means or why is it happening. But some conclusions that we were able to draw from this is that WGS is able to offer a slightly increased yield in clinical samples, that the prevalence of heteroresistance is rather low, and when it does happen, it seems to have limited clinical relevance, and that there's no significant evidence of culture bias in our data set. Of course, it's important to consider that we are working with the lineages of tuberculosis which are less susceptible to culture bias, so that's something to bear in mind. And we would also think that much larger studies are needed, which incorporate additional analysis, including things such as cost effectiveness. I would like to end by highlighting the collaborations that have made this study possible, especially with Rob Warren's group here at the University of Stellenbosch, but also with our partners such as the University of the Free State, the Orem Institute, NHLS Bloemfontein, our funders, and of course, PHA4GE for allowing us to share a little bit of our work with you today. Thank you very much.